Designer babies. A huge deal in science fiction for years as fiction explored the idea of babies and changing our offspring to make them exactly the way we want. But what if I told you this isn't just science fiction? Today we are going to be talking about genetic engineering and how it could affect what our babies look like in the future. It all started back in 1987 with the discovery of CRISPR, which is also known as short, clustered, regularly, interspaced, short palindromic repeats. CRISPR are repeating DNA sequences in the genome of prokaryotes, such as bacteria. CRISPR was discovered by Yoshimo Ishino and his team, who accidentally cloned an unusual series of repeated sequences interspaced with spacer sequences, while analyzing a gene responsible for the conversion of alkaline phosphate in E. coli. But this was just the start of CRISPR's journey. Let's fast forward to 2012 when CRISPR-Cas9 was invented. CRISPR-Cas9 was the start of being able to genetically modify humans. Cas9 enabled geneticists and medical researchers to edit parts of the genome by removing, adding, or altering sections of the DNA sequence. Cas9 acts as a pair of molecular scissors that can cut the two strands of DNA at a specific location in the genome so that bits of DNA can be added or removed. So what does this mean for the future of humans? Beyond research, it may soon be feasible to use germline genetic engineering in human reproduction. The most obvious clinical use of germline gene editing would be to correct mutations associated with fatal single gene disorders such as Tay-Sachs disease, Duchenne muscular dystrophy, cystic fibrosis, and spinal muscular atrophy. These conditions are caused by well-understood genetic mechanisms and can reduce life expectancy by decades. In these cases, using germline gene editing will be the only way that individuals can avoid serious diseases in their children. In the far future, perhaps in a few decades, we may be in the position to use embryonic gene editing to target other causes of death. Roughly 30% of all deaths worldwide are due to chronic illnesses, such as heart disease, cancer, and diabetes, in those under 70. Many billions of dollars are spent each year trying to develop new treatments to these disorders and reduce their impact on mortality. We know that these chronic diseases is affected by our genetic makeup. For example, genome-wide association studies have identified at least 44 genes involved with diabetes, 35 genes involved with coronary artery disease, and over 300 genes involved in common cancer. As we understand more about genetics and more about the etiology of these disorders, it will be possible to reduce our susceptibility to these diseases. The ability of germline gene editing to target multiple genes simultaneously means it could potentially be used to reduce the incidence of these disorders. As we can see, while science fiction may have painted gene editing in a scary light, this very unique approach to science could be used to save multiple lives and help the world. So the next time someone talks about gene editing, know it's not as scary as it seems.